Hello everyone, welcome into the ITLS Academy. I am Dr. Himani, Senior Research Consultant in ITLS Academy. Hopefully you all enjoying our online training program. So today we are going to discuss about medical biotechnology is advancing modern healthcare. So now first we will be go for understanding what is the biotechnology. Biotechnology contributes much towards the growing public and global health need. It has revolutionized mankind since its existence. Any technology application that use biological symptoms, living organisms or derivatives thereof to make or modify products or process for specific use. It provides effective diagnostic, prevention, and treatment measures include production of novel drugs and recombinant vaccine. It gives effective drug delivery approaches, new methods for therapeutics, nutritionally enriched and genetically modified crops, and efficient methods for environmental cleanup. Health, life quality, and expectancy of life have been increased worldwide through the services provided by the biotechnology. It is a toolbox filled with many different kind of living cells and their components, molecules, and the many different ways to use them. There are millions of different species of plant, animals, and microorganisms in the world. So now I hope you will be understand what is the term of biotechnology. Next, we are going to discuss about the history of biotechnology. Here, the history of biotechnology is divided into the three stages. First one is ancient biotechnology, second one is classical biotechnology, and third one is modern biotechnology. Now, first we will be discuss about the ancient biotechnology. It can be referred to all development or discoveries that took place before the 80s. These were all mainly to fulfill the basic need of humans such as food, cattle, or shelters. Early hunting was a common practice for food gathering, and eating raw meat was ordinary. It took time to understand the importance of fire, clean water, definitely agriculture also. With agriculture came the domestication of animals and food like cheese, curd, and others. Possibly, cheese was the first product of biotechnology as it was prepared by adding an enzyme called rennet to our sour milk. So now, second stage of biotechnology is classical biotechnology. Classical biotechnology we can define as a now the development and discoveries in the biotechnology were at it it exponential phases. Morgan in 1926 gave the theory of gene who showed the world that chromosomes play an essential role in inheritance by using food file. It was Johnson's who coined the term gene and described it as a carrier of heredity. Now, third stage is modern biotechnology. After the Second World War, some very crucial and important discoveries were made, which set the stage of modern biotechnology. In 1975, Fowler and Milton came up with the concept of cytoplasmic hybridization. They also produced first monoclonal antibodies which revolutionized the diagnostic world. Dr. Hargobind Khurana was successful in synthesized DNA in test tubes, whereas love of chemistry for K. Mullins led to the development of PCR, which added volume, we can say which added value to Khurana's work by amplifying DNA 1000 times more than the actual amount. So now here we are going for classifications of biotechnology. The scope of biotechnology in our world has derived a need to classify biotech based or on some common features or their final purpose. There are some of the main area of biotechnology using a color classification. So first one is red biotechnology. We can say we can classify biotechnology in the four part. First one is red biotechnology. White. Second one was the white biotechnology, and third green biotechnology, and fourth classification is blue biotechnology. Now we are going to discuss about the red biotechnology. Red biotechnology, or we can call it as biopharma, bring together all those biotechnology uses connected to medicine and veterinary product. Red biotechnology include producing vaccine and antibiotics, developing new drugs, molecules, diagnostics techniques, regenerative therapeutics, and the development of genetic engineering to cure disease through genetic manipulation. Now, we are going to discuss about the white biotechnology. In the white biotechnology, we can say that white biotechnology related to industrial biotechnology. White biotechnology play a special attention to designing low resources consuming process and products, making them more energy efficiency and less polluting than traditional ones. So now the turn of green biotechnology. Green green biotechnology is focused. Sorry, green biotechnology is focused on agriculture. That's why you can understand the name is come as a green biotechnology because it just related to agriculture. So green biotechnology approaches and applications include creating new plant variants of agriculture interest, producing biofertilizers and biopesticides. This area of biotech is based exclusively on the transgenic, or we can say genetic modification. Now the term is blue biotechnology. Blue biotechnology based on exploitation of marine resources to create product and application of industrial interest, taking into taking into account that sea present the greatest biodiversity. There is a potentially a hug range of sector to benefit from the use of this kind of biotechnology. Now we are going to discuss about the what is medical biotechnology. Medical biotechnology is a branch of medicine that use living cells and cell material to research and then produce pharmaceutical and diagnosis products. These products help treat and prevent diseases, from the Ebola vaccine to mapping human DNA to agricultural impacts. medical biotechnology is making hug advantage and helping millions of people. Some of the most recent use of biotechnology is work in genetic testing, drug development, treatment, and artificial tissue growth. With the many advancements in medical biotechnology, there are new concerns that arises. Now, here is the four types of application of medical biotechnology, which are they are like pharmacology, genetics, therapy, and stem cells and tissue engineering. Now we are going to discuss about the pharmacology. Pharmacology is the branch of medicine and biology concerned with the study of drug action, where a drug can be broadly defined as any man-made, natural, or endogenous molecule which exerts a biochemical and physiological effects on the cell, tissue, organ, or organism. Study of the interaction that occur between a living organism and chemical that affect normal or abnormal biochemical function. Use of pharmacology. Uh, we can use of pharmacology in the four sections. First one is insulin production. Second one is human growth hormone. Third one is human blood clotting factor, and last one is monoclonal antibodies. So now we are going to discuss about the insulin production. Production of genetically engineered human insulin was one of the first breakthroughs of biotechnology in the pharmaceutical industry. Insulin was first produced in Escherichia coli through recombinant DNA technology in 1978. The the human gene for insulin is placed into bacteria, are cultured and allowed to produce insulin, which is collected, purified, and sold to diabetic worldwide. So now, human growth hormone. Production of human growth hormone was first done in 1979 using recombinant DNA technology. Scientists produced human growth hormone by inserting DNA coding for human growth hormone into a plasmid that was implanted in Escherichia coli bacteria. This gene that was inserted into the plasmid was created by reverse transcription of the mRNA found in pituitary gland to complementary DNA. Prior to this development, human growth hormone was extracted from the pituitary gland of Cadbury as animal growth hormone have no therapeutic value in humans. Now we are going to discuss of human blood clotting factor. So clotting factor isolate from blood are used to treat some hereditary bleeding disorders such as hemophilia. Human blood clotting factor were produced from donated blood that was in, inadequately screened for HIV. Thus, HIV infection posed a significant danger to patients with hemophilia who received human blood clotting factors. In the biotechnology, maybe we can say like that in this biotechnological procedure, the human gene that codes for the blood clotting protein in transferred to hamster cell grown in tissue culture, which then produce factor 8 for use by hemophilia. Now we are going to discuss about the monoclonal antibody. Monoclonal antibodies are antibodies that are made by identical immune cells that are all cloned of a unique parent cell. Remember, antibodies are specific proteins that target pathogens invading our body. Monoclonal antibodies can have monovalent affinity in that they bind to the same epitope. Yeah, definitely we can say like that the part of an antigen that is recognized by the antibody. So now the advantage of monoclonal antibodies. This technology is used primarily to fight off cancer cell, as these monoclonal antibodies can be trained to target markers that show up on cancer cell. Monoclonal antibodies are administered through a vein, or maybe we can say like that intravenously. How often you undergo monoclonal antibodies treatment depends on your cancer and the drug you are receiving. Some monoclonal antibodies drugs may be used in the combination with other treatments such as chemotherapy or hormone therapy. The antibodies will then destroy the cancer cell and go looking for more. Now we are going for discuss about the gene therapy. Gene therapy is an experimental technique that uses gene to treat or prevent diseases. Gene therapy is the use of DNA as a pharmaceutical agent to treat disease. Although gene therapy is a promising treatment option for a number of diseases, including inherited disorders, some types of cancer, and certain viral infection. Gene therapy is currently being tested only for disease that have no other cures. It derives its name from the idea that DNA can be used to supplement or alter gene within a individual cells as a therapy to treat disease. The most common form of gene therapy involved using DNA that encodes a functional therapeutics gene to replace a mutant gene. In the future, these techniques may allow doctors to treat a disorder by inserting a gene into a patient cells instead of using drug or surgery. Now we are going to discuss about the stem cells. A stem cell is a cell that has the potential to become any cell type in the human body. Here you can see like that stem cells provide new cells for the body as it grow and replace specialized cells that are damaged or lost. Here you can see like that in this there is a single cell that can be replaced in itself, or it can be replaced in different type of cells. It could be a neuron cells, blood cells, uh, maybe so many type of cells. It could be differentiate its own the way. So there is a great interest in stem cells because they have potential in the development of therapies of replacing defective or damaged cell resulting from a variety of disorders and injuries such as Parkinson's disease, heart disease, and diabetes. They have two unique properties that enable them to do this. First one is they can divide over and over again to produce new cells. As they divide they can change into the other type of cells that make up the body. So now, types of stem cells. Here is the two type of stem cells. First one is embryonic stem cells and second is adult stem cells. The embryonic stem cells used in research today come from unused embryos. These result from an in vitro fertilization procedure. These embryonic stem cells are pluripotent. This means that they can turn into more than one type of cell. Now, what is the adult stem cells? Adult stem cells supply new cells as an organism grows and to replace cells that get damaged. Adult stem cells are said to be multipotent, which means they can only change into some cells in the body and not any cells. Now we should discuss about the stem cells in medicine, because stem cells have a very important role in the medicines industry. So the only stem cells now used to treat disease are hematopoietic stem cells. These are the blood cells forming adult stem cells found in bone marrow. So you can understand the stem cells derived from two things. First one is from the blood and second one from the bone marrow. These cells are used in procedures such as bone marrow transplant. These help people with cancer make new blood cells after their own hematopoietic stem cells have been killed by radiation therapy and chemotherapy. Stem cells may help your health in the future in many ways and through many new treatments. Researchers think that stem cells will be used to help create new tissue. For example, one day healthcare providers may be able to treat people with chronic heart disease. They can do this by growing healthy heart muscle cells in a lab and transplanting them into a damaged heart. Other treatment could target illness such as type 1 diabetes, spinal cord injury, Alzheimer's disease, and so many types of arthritis diseases. New medicines could also be tested on cells made from different type of stem cells. Now, main thing is tissue engineering. We are going to discuss about the tissue engineering. Tissue engineering evolved from the field of biomaterial development and refer to the practice of combining scaffold cells and biological active molecules into functional tissue. The goal of tissue engineering is to assemble functional constructs that restore, maintain, or improve damaged tissue or whole organ. The focus of tissue engineering is in restoring, maintaining, and improving function of damaged tissue and organs. Researchers combine the principle of engineering and life sciences to find synthetic and naturally derived materials to create functional tissue in the lab and then apply them to the affected areas. Artificial skin and cartilage are examples of engineered tissue that have been approved by the FDA. However, currently they have limited use in human patients. So here we can understand how we can get an engineered tissue. Engineered tissue is the combination of biomaterial scaffolds, cells. In cells, we can use stem cells, fibroblast cells, and different kind of cells we can use it. And growth factor is also important for the making a engineered tissue. So we can say like that engineered tissue is the combination of biomaterial scaffolds, cells, and growth factors. Tissue engineering thread of the cells signals and the scaffolds which act as a template for tissue formation by allowing cells to migrate, adherent, and produce tissue. Tissue engineering field relies extensively on the use of porous 3D scaffold to provide the appropriate environment for the regeneration of tissue and organs. These scaffolds essentially act as a template for tissue formation and are typically seeded with cells and occasionally growth factor or subjected to biophysical stimuli in the form of bioreactor, a device or system which applies different type of mechanical or chemical stimuli to cell. So these cell seeded scaffolds are either cultured in vitro to synthesized tissues which can then be implanted into an injured site, or are implanted directly into the injured site using the body's own system, while regeneration of tissue or organs is induced in vivo. So now next is scope of biotechnology. This is the main thing which one we are going for discussing about scope of the biotechnology. Biotechnology is applied to variant fields and many industries such as food, pharmaceuticals, medicine, agriculture. It has its scope in both research and engineering. Genetic engineering has helped in the production of therapeutic proteins as well as biological organisms. Biotechnology has made major advances in molecular biology and industrial biotechnology. The scope of biotechnology is extended to various branches of biology. Some of these include tissue culture, development of transgenic plants and animals, development of antibiotics. USA itself has established more than 200 companies such as Biogen, Hybridtech, etc. So, all covered by me. Hopefully, you all enjoying this lecture. Thank you.